We're going to discuss some carboxylic acid derivatives, meaning that they were probably generated from carboxylic acids most of the time. And this time we're going to start talking about amides, and that's where we're going to begin looking at when we've, as I mentioned earlier, we can have these substitution reactions where we pop off that hydrogen and we now replace it with nitrogen groups. So we can have different types of amides, primary, secondary, and tertiary amides, right? Where we have either it's at the end and we just have two hydrogens off of the nitrogen. We have one hydrogen or an R group or a chain coming off, so secondary amide. This is more what we're going to see in amino acids. And then the tertiary amide, where we have maybe two chains coming off of that group. Unsubstituted amides can form three strong hydrogen bonds, and that's because they'll have multiple areas where they can interact with hydrogen. The nitrogen can bond to another hydrogen, and these two hydrogens off of that nitrogen can bond to two other nitrogens or oxygens. So they produce a lot of different places where they can interact. More substitution, the less hydrogen bonding, right? Because we have now this substitution, only this hydrogen can hydrogen bond. We can't have two additional spots because the carbon's not going to hydrogen bond. And then with this one, this actually can't hydrogen bond with itself, this compound, but it can be bound with by, for example, with water. So it can be involved in a hydrogen bonding with another molecule, but it itself cannot hydrogen bond. So some differences between, for example, carboxylic acids. You'll notice here with this carboxylic acid, we have a hydrogen bonding between one carboxylic acid end and another carboxylic acid end. These can interact with and hydrogen bond with each other. Also, you'll notice amides, that nitrogen and that hydrogen can hydrogen bond to that carbonyl oxygen. And so this can happen in many different ways here. So these two, both carboxylic acids and amides, can hydrogen bond with itself, with each other, in a pure solution. Now, you'll notice here, there is not that ability for esters to do that. So it generally means that carboxylic acids and amides have higher boiling points because of that intermolecular force that's possible there. Amides are generally not basic, which means they're not going to take on a hydrogen form. So they will generally, because of this positive electropositive charge here, the positive end of the carbonyl group is going to attract these unshared electrons. So they're not going to be available for bonding to a hydrogen, for example, that's kind of floating around in solution. Not going to happen because these electrons are flowing closer to that carbon there. So this is the same type of substitution reaction that we've seen in the past that we were looking at for esters as well. We're going to form water here. This OH and this H are going to come over here and form water. So we have, remember, nitrogen's more electronegative than hydrogen. So this electronegative element is going to be attracted to that hydrogen. This electronegative element is going to be attracted to that carbon that's going to be partially positive. And then we're going to basically pop off that hydrogen, that OH and that hydrogen, and we're going to form water. And then we're going to have that primary amide. So this is the way that we can form unsubstituted amides, because this really, the only way to do that is with ammonia, because we have to have that attached to another two hydrogen molecules. So that's the easiest way to do that. If we're going to have the substituted amides, we're going to generally have primary, secondary amides as that replacing group. So it's going to probably be attached to something already. Here's methylamine, for example. So that group is going to come along with that nitrogen and form a longer chain here, as we can see in this example. So here we're going to, I want you to take a few seconds here to look at these reactants. So here I want you to look at these reactants, and I want you to pause this after you have solved what you believe this would result in. And this is the solution. We have this nitrogen is going to be partially negative, and that's going to be attracted to this partially positive center on the carboxylic acid. That's going to come into there. These electrons are going to pop onto that hydrogen, and one of these are going to be used to form that water molecule. So that's why we only end up with NH, and then that methyl ethyl group is on the other side there. The reverse reaction can also occur, although substitution reaction, like the others we've been showing, there's more specific names given to many of these reactions. So this is still a reverse reaction, still a substitution reaction, but because we're adding water, we call this a hydrolysis reaction. So there are specific names that can be given to some of these reactions. The general kind of simple name for this is a substitution reaction. But when water is involved, we can get more specific and say it's a hydrolysis substitution reaction. And just to kind of note to that, it's usually we're using water. It's like water lysis. We're using water to break this bond, right? Essentially, it'd form this new bond, but it's really a substitution reaction. But you can say it's a lysis because we're cutting this longer chain up. So this was the answer to the last one. All right, so now what I'd like you to do is I would like you to look at these chemicals, and I want you to order them from the lowest to the highest boiling point, and then think about why. So why did you arrange them in that way? What was your reasoning? So unpause after you have a chance to do that. All right, so we're going to look at this, and something that I noticed right away, I'm going to be looking for which compounds have the ability to hydrogen bond. Now, this oxygen to hydrogen, that will be able to hydrogen bond. This oxygen is attached to a carbon and another carbon, so is this one. No hydrogen bonding here, so I'd expect this to have one of the lower boiling points. The same thing here. We have a nitrogen with two regions that they can hydrogen bond. So I would think that this, because this has two hydrogen bonding hydrogens, and this only has one, that this would probably be a little higher than this one. And the difference between these two is the chain length. So then, even though it's a little bit longer, right, that gives it more ability to do the dipole-dipole interaction, van der Waals, whatever you want to call it, right? So I would say this is going to be the highest, next highest, third highest, and then finally the least highest because it doesn't have the ability to hydrogen bond. So that concludes our discussion on amides. We're going to talk about phosphoric acids now, another derivative of a carboxylic acid. And you'll notice that there's a big similarity between carboxylic acid and phosphoric acids. The difference obviously being that we have additional hydroxide groups coming off of there. Phosphate is a little lower on the periodic table. It has the capacity to expand into the d orbitals and hybridize its orbitals a little more to accommodate more electrons around this larger molecule. So it's a little different than some of the other organic compounds we've looked at in the past. So there are 3 ionizable hydrogen atoms in the red. So these we can have, that's why it's a phosphoric acid. It acts like an acid. It's actually able to release those as an acid in those different forms. Just like carboxylic acids, these phosphoric acids are able to form phosphate esters. So we can have, for example, this primary alcohol. It is able to, this phosphate is going to be partially positive because of all of these bonds with oxygen. So a negatively charged oxygen may be attracted to that region. It can pop off one of these hydroxide groups. It could be this one, this one, this one. It doesn't really matter, right? It's the same, going to be the same anyway. And we can end up with a methyl phosphate. So it's kind of like an ester but it's called a phosphate ester because we have a phosphate here instead of a carbon. Interestingly, phosphoric acids commonly react with these acids, or weak acids, right? We have threonine, which is an amino acid, and it's the side chain of the amino acid it's going to have access to. So threonine is this OH group, so we can have that phosphoric acid interact here. Another hydroxide group here that it can interact with, and serine. So threonine, tyrosine, and serine. These are three amino acids that commonly get phosphorylated in cells, in our bodies, and in our proteins. So acids can form esters between themselves as well. So we've heard of, right, ATP, AMP, ADP, monophosphate, diphosphate, triphosphate. Here's triphosphoric acid. And you might have also heard of diester bonds, diester. There's two of them, right? 1, 2, we form this diester diphosphate bonds. So that's what we're talking about when we say that diester bond. If you've heard that in the past, that's because we have these diesters. They're all often, and I just mentioned this, they're often provided by ATP or ADP, AMP, monophosphate, that is also accompanied by the release of energy. So when we break those bonds, then we generally can generate energy from that. We can harness energy. So these have high chemical potential energy, and these have lower chemical potential energy, and then we can harness that through other means in our bodies and in the reactions we're going to look at in the future here. We're also now going to begin looking at ethers. Ethers are here. We can have, when we don't have, now we're losing that carbonyl group. Now we're just looking at chains of carbon where an oxygen is replacing, maybe kind of, you can look at it as replacing a carbon in here. We're not going to have any oxygens attached to these. And so this is referred to also as an alkoxy group. So if we're looking at, for example, if an oxygen with a methyl group on the end, it's called a methoxy group. Oxygen with an ethyl group on the end, ethoxy group. So these are used when the compound has other functional groups. So in your homework, I believe you probably noticed this. We have O for the position, right? We have ortho and para and meta positions. And this is the O-methoxy phenol, because this is a phenol functional group. So you don't necessarily have to memorize that this is a phenol group. But what I do want you to know is that when you have a benzene ring and you have other things hanging off of it, sometimes there are common names that are used in IUPAC naming, like a phenol group. And basically what I'm expecting you to do is be able to look that information up. So most ethers are non-reactive with most reagents except for oxygen, and they're flammable. They're not common biochemicals. You don't see this ether very often in biomolecules because they are somewhat unreactive. So what I want you to do is right now look at these. I want you to identify these following compounds. Which ones are carboxylic acids, amides, esters, ethers, none of these. What are they? So take some time, draw these out, define what they are, and I'm not going to go over the answers with these, but if you want to come and see me in my office hours or discuss these further, if you're not sure how to draw these out, I recommend coming to me or talking to one of your classmates to figure this out, because this is what I want you to be able to do on an exam, is to identify these, be able to draw these out, draw out their structural formula, and name them. I'm going to now go into amines and talk about some of the properties of amines. They're major components of proteins. They're volatile amines. When they get smaller, they can kind of aerosolize and they can smell really bad. So a lot of times you'll have primary amines, if you smell something really nasty in a chemistry lab, they can have the scent of rotten fish or decaying meat. Putrescine is a common additive to many cell cultures because as you can imagine, putrescine sounds putrid, but it's a source of nitrogen. And nitrogen is an essential component of the human body, and because they're a component of proteins, which are enzymes. So we rely on these nitrogen sources, but that doesn't mean that they smell great. Can have ammonia, obviously, is a nitrogen that has 3 hydrogens attached to it. Just like carbon naming system, as I mentioned a few lectures ago, we have the nitrogen, we have a primary amine, which is attached to 1 chain of some sort. A secondary amine is attached to 2, and a tertiary amine is attached to 3. You can have a quaternary amine as well. Here, I'm showing you here. There's always going to be positively charged, and the reason for that is that these lone pair of electrons, right? Normally nitrogen has 5 electrons, and it's got 1 it's sharing here, 1 it's sharing, 1 here, 1 here, and 1 here. So it's 1, 2, 3, 4, 5. 5 electrons. 1, 2, 3, 4, 5. And if it's going to start sharing another one of those, right, it really only then has 4 of its own. So it's lost an electron. Electrons are negatively charged. If it lost one of those, it's going to take on a positive charge. So quaternary nitrogens, when they're attached to 3 other things, they're going to be positively charged because of that. Okay, so were you paying attention to what I just told you? How do you name this? What is this? A primary, secondary, or tertiary amine? So this is going to be a primary amine. It might have tricked you because you're looking at this carbon and it's a secondary carbon, but that's not how we name nitrogens, right? If this was an alcohol, it would be a secondary alcohol, but amines, we're looking at how many carbons the nitrogen's attached to. So it's actually going to be a primary amine. How about this one? Take a second and unpause when you figured it out. Right, so this one is going to be this, we're looking at this nitrogen. It should give you a clue right here. There's a positive charge associated with it. Oops, sorry. And that should mean that you should automatically know it's quaternary. But also if we have these methyl groups, we have 3 methyl groups. And that means it's attached to 4 things because it's also attached to this carbon. So if you were to draw this structure out, you'd see that this is attached to 4 things. So that means it's a quaternary amine. So I'm going to just go over quickly naming primary amines, and it's not something that you need to necessarily memorize for exams, but it just, again, to make sure that you can recognize this. If you see the name, you should be able to say, okay, there's an amine in here. So the parent group is the amine. So that is going to go on to the end, depending on what other functional groups are found there. And you're going to say methyl, ethyl, so that chain is going to be ethylamine, isopropyl because methyl ethyl propyl, and it's on that second. We have that special terminology for these kind of 3 simple carbon groups that have that secondary carbon attached to the amine. So isopropylamine could have, and then also cyclohexylamine. So we generally put that that chain in front. If it's a primary amine, we can put that chain in front and then just put the amine on the end. Things get a little more difficult when we have secondary and tertiary amines. And usually we'll do something like the dipropyl, methyl ethyl propyl. So we have dipropylamine or triethylamine. So if they're both the same on both sides, we can do that. Otherwise, we get to once we get to more complicated things that have different chains on both sides, we start naming this instead of numbering the chain as like 1, 2, 3, 4, 5. We just tell people what is coming off the nitrogen. So there's an ethyl group off the nitrogen, so N-ethyl propylamine. So we just say this is the substituent and this is the main chain. And here these are 2 substituents, so it's NN, just like you might write 1,1-dimethyl, but instead it's not on a 1 carbon, it's on the N, right? It's on the amine. So NN-dimethyl, and this is the long chain, so it's a propylamine functional group. So again, you don't have to memorize this, but if you see these kind of words, it shouldn't throw you off and send you into panic, right? So it just means that it's just telling you where that substituent group is attached here. Common names for some of these benzene rings, right? We have common names for some of these. Aniline is a common name for the benzene ring with the primary nitrogen. And then we know these amino acids are also amine, right? Amino acids, not necessarily going to name it through IUPAC naming system, but it is an amine with carboxylic acid functional group. And I want to show you some common nitrogen containing compounds that are common in biochemistry. Pyrrolidine in nicotine and imidazole looks very similar to histamine, and this can commonly be used in purification techniques for different H-tagged proteins. Purine rings should look familiar to you from DNA. Pyrimidine as well. It's also present in vitamin D or vitamin B. Quinoline, antibacterial agent, an indole ring, pyridine, and piperidine. Different nitrogen-containing compounds in various drugs. These are also commonly occurring in many different types of plants. Morphine, we have nitrogen-based compounds here, heroin, codeine, and many of these alkaloids are physiologically active in humans. Caffeines and nicotine, for example, are stimulants. Some are pain relievers. What do you notice about the differences between these drugs? If you look at these, there's not a very big difference, right? We have over here, we have an alcohol, and now we have an ester, and over here we have ether and alcohol down here. So there's not that much of a difference between morphine, heroin, and codeine. And that's why all of these things have the very similar addictive properties, as you may expect. So as you might also expect, as similar to the amides, amines also have the ability to hydrogen bond because of the hydrogens attached to them, at least depending on if it's a primary or secondary amine. Obviously, then we're able to, because of this, have a slightly higher boiling point, right? If you're looking at butane, if you substitute nitrogen on the end, all of a sudden it goes up 50 degrees, even though the molecular weight is pretty much the same. Not quite as good as hydroxide, right, where we can go up to propanol as a boiling point of 97 degrees, but these do help with the hydrogen bonding. And that's partially because that oxygen is more electronegative than that nitrogen as well. That's why it's the higher boiling point. Some more heterocyclic nitrogen compounds that are common in DNA, right? Deoxyribonucleic acid. So we're talking about base pairs. We're talking about bases because we have these nitrogens that act as, normally nitrogen-containing compounds act as hydrogen acceptors and therefore bases, and that's why they're called base pairs. So the lone electron pairs on amines cause them to be weak bases, as I was just mentioning, and the electron pair donors by forming with a bond with hydrogen ions, and then they can form an acid in water. So here we have that lone pair. It may accept hydrogen from that water, for example, and then this can act as an acid. So I want you to sit here and think about write the balanced equations for the following reactions. What is going to happen here in both of these situations? So we're going to take NH3. Let's start over here. We know that these electrons are going to be interested in interacting potentially. We have a partial negative and partial positive here. Partial negative, partial positive, remember, based on the electronegativities. So the oxygen and the nitrogen aren't necessarily going to want to interact, but the electrons here and this hydrogen may want to. And so these electrons will flow towards the OH group, and these electrons will bond to that hydrogen. And since they're now, this nitrogen is going to lose one of these electrons of that hydrogen, we know that quaternary, or that when nitrogens are attached to 4 things, it's going to take on a positive charge. And we're also left with an OH group that is negatively charged. So this is the balance equation for ammonia and water. Now this next one is a trick question. So let me pause this and erase some of this other stuff. This nitrogen, I'm switching up the color for fun, is now attached to 4 different compounds here. And it's going to be positively charged. This OH group, it's floating in solution, negatively charged. Now, they might be wanting to interact, but there's nowhere for these nitrogens or these electrons to flow to. We cannot add another bond to the nitrogen because all of those electrons, all of those spaces are taken up. So actually no reaction forms because they can't form a covalent bond. So it was a trick question. Sorry if that was really confusing to you. They can form a salt. They can kind of associate with each other, but no covalent bonds are going to be formed because there's nowhere for those electrons in the oxygen to flow towards in the nitrogen. So it's not going to actually kick off a CH3 group. We're not going to have that occur because generally we don't have negatively charged carbons floating around in solutions. So that's not a common chemical reaction that we've talked about today. We can form, though, however, amine salts. So an ammonium salt, again, here's a definition, is the ionic compound composed of a ammonium cation and an anion and an amine salt. And so generally we do this a lot of times in order to make things more water soluble. So you'll make a salt. So you might see this on some other drugs. I'll show you in a second here an example of that. So amines, generally positively charged amine groups that are formed by the addition of an acid, are called ammonium ions. And so these amines generally exist as ammonium ions in the aqueous environment, in the blood and other bodily fluids, because otherwise they're not as soluble in water. So histamine is this positively charged group here. Serotonin is a neurotransmitter, generally have positively charged ammonium ions. And these are really important. Most of these times, these molecules are really oily liquids, and they're difficult to formulate in that state, in that functional state, if they're not amine salts. So they're converted into those salts so that they can be water soluble, and obviously that's important if we're going to eat them or take them into our bodies. I'd like you to look at this chemical reaction with Sudafed, and I want you to write out the product of the reaction when hydrochloric acid is reacted with pseudoephedrine. This is the solution, and you'll notice that the hydrochloric acid is going to interact here with the pseudoephedrine to make a acid salt. That nitrogen there is going to interact with the hydrogen in the hydrochloric acid, and then we are going to then have that positive charge on the nitrogen because it's attached to 1, 2, 3, 4 things. And then we're going to have that negatively charged ion also associated with it to make that salt compound that's going to be more soluble in our bodies. So now which is going to be more soluble in water here? The compound on the left or the compound on the right? So phenylephrine hydrochloride is going to have that positive charge because we've interacted hydrochloric acid with it to make a amine salt, and it is going to be the more water soluble.